Good morning, students. Today we are going to learn about a very interesting topic, that is acid base indicators. So, let me ask you a simple question. If I will give you a colorless liquid, how will you identify whether it is a base or it is an acid? Take your time and answer. Most of you will answer by the help of litmus or turmeric or something which will change its color. By the help of that, you will identify whether it is acid or base. So, what is that litmus? Litmus is also known as what? Okay. So, from this, our topic of discussion comes, that is indicator. So, what are acid base indicators? Or simple, we can say indicators. Indicators or acid base indicators are the substances that change color when added to acids or bases. They help us to identify the nature of the solution, whether it is acidic, whether it is basic, or whether it is neither acidic nor basic, that is neutral. Most of the indicators, they change their color when added to an acid or added to a base. Color change happens. Coming to types of indicators, there are mainly three types of indicators. The first one is natural indicator, second one is synthetic or man-made or artificial indicators, third one is olfactory indicators. So let's discuss about the natural indicators. Okay, natural indicators means the indicators which are available in nature, which are not made by human, which are already available, which we can collect from the nature. Okay, you have already read about these natural indicators in your lower classes. You have read about litmus, you have read about turmeric, you have read about the hibiscus, China rose. You have read about a lot of natural indicators in your lower classes, junior classes. Let's discuss about the litmus, the first natural indicator, which is very famous. That is litmus paper. Litmus is available in solution form and it is also available in paper form. Most of you have used the litmus paper. One is blue litmus paper, another is red litmus paper. So let's know something more about the litmus. So litmus solution is a purple dye. Talking about the litmus solution, it is a purple dye which is extracted from lichen. From lichen is a tree available in nature. So litmus solution is extracted from lichen, a plant belonging to the division Thallophyta, and is commonly used as an indicator. When the litmus solution is neither acidic nor basic, its color is purple. There are many other natural materials or natural indicators like red cabbage leaves, turmeric, colored petals of some flowers such as hydrangea, petunia, geranium, which indicate the presence of acid or base in a solution. These are called acid-base natural indicators or sometimes simply indicator. So there are a lot of examples of natural indicators. The famous one is litmus solution. See, in the figure, there are two types of litmus papers. One is blue litmus paper, another is red litmus paper. See, when we put the blue litmus in the acidic solution, it turns to red color. So from this we know that acid turns blue litmus to red. Like that, when the red litmus paper is put in the basic solution, it changes its color to blue. So from this we know that the base changes red litmus to blue. Simple. This is about acid turns blue litmus to red and base turns red litmus to blue. This is all about litmus paper. See another figure. In neutral solution, red litmus remains red, blue litmus remains blue. In acid, red litmus remains red, but blue litmus turns red. And in base, the red litmus turns blue and blue litmus remains blue. Okay. Talking about the China rose indicator. China rose is otherwise known as hibiscus. China rose petals, if the China rose petals will be kept in warm water, then a colored solution is obtained. And that colored solution of China rose is used as an indicator to identify the acidic or basic nature of substances. If we put acidic solution inside the China rose solution, then the color will be changed to magenta or dark pink. If we put basic solution inside the China rose indicator solution, then the color will be changed to green. Okay. And in neutral solution, the China rose indicator solution remains light pink. So this is all about the China rose indicator. Talking about the turmeric indicator, the turmeric remains yellow. Turmeric is generally yellow in color. So the turmeric remains yellow in the acidic solution, but in basic solution it is changes to reddish brown. This is about turmeric. So these are the famous natural indicators. Coming to the artificial indicators, which are very important. The artificial or man-made indicators, which are made by human in the laboratory. There are main two important artificial indicators. One is phenolphthalein and the second one is methyl orange. First we will talk about the phenolphthalein indicator. See, the phenolphthalein indicator is in general, the phenolphthalein indicator is colorless. When you will see the phenolphthalein indicator in lab, it is colorless. Okay. But when we put the phenolphthalein indicator inside a basic solution of pH 8 to 10, then it will turn to pink color. Okay. When we put phenolphthalein inside an acidic solution of pH range 0 to 3, that means highly acidic solution, the color will change to orange color. If we put phenolphthalein indicator inside a highly basic solution of pH range 11 to 14, then it will again remain colorless. So these are the color changes of phenolphthalein indicator in different pH solutions. You have to remember this. These are observed experimentally. So again, the color of the phenolphthalein in between the pH 8.3 to 10 is pink to fuchsia, and the color of the phenolphthalein begins to fade and become colorless in a strongly basic solution, that is pH greater than 13. And the color of the phenolphthalein is orange between the pH range 0 to 3, and it again remains colorless between the pH 4 to 7. So this is all about phenolphthalein indicator, and this phenolphthalein indicator is used in the acid base titration. Coming to methyl orange indicator, let's observe its color change. In neutral pH, in neutral pH, the color of the methyl orange indicator is yellow, and in alkaline pH also the color of the methyl orange indicator is yellow. But in acidic pH, the color of the methyl orange indicator is red to orange. Okay. When the pH is less than 3.1, the color of the methyl orange is red. When the pH is 3.1 to 4.4, the color becomes orange. When the pH becomes greater than 4.4, the color changes to yellow. And above that, in the basic solution also, the color of the methyl orange becomes yellow. So this is the color change of methyl orange indicator. You have to remember. See, this figure you are looking in the screen, looking on the screen is a pH paper. You all know about the pH scale. In the pH scale, the range is from 0 to 14. A neutral solution has a pH of 7, and below 7, if the pH is less than 7, then the solution is acidic pH. If the pH is greater than 7, that means between 7 to 14, then that is basic pH. The solution is basic. So this is the pH scale. So based on the pH scale, there is a universal indicator, that is pH paper. The pH paper is known as the universal indicator. So the figure of pH paper is on the screen. When we put the pH paper in any solution, like in acidic, basic or neutral, then its color changes, and after changing the color, you have to match the color with the color in the pH paper, and then you can say what is the pH of the solution. This is the procedure of using pH paper and knowing pH of a solution using pH paper. Okay. See, this is an example. A student put the pH paper in a solution, the color changes to green, and he matches the green with the pH paper. The green color pH range is 10, so the solution has a pH of 10. Okay. See, this is the different. There are different colors of different pH in the pH paper. See, this is another student take a pH paper, put inside a solution, then the color changes to blue, then he matches the color with the pH paper, then he identifies the color and the pH. Okay. And nowadays digital pH meter is available. pH meter is an instrument which gives exactly accurate reading. What is the accurate pH of a solution? That is given by the pH meter. See, on the screen, the instrument is shown. This is the digital pH meter available nowadays. It will, if we will put the pH meter inside a solution, then it will show the pH of that solution immediately. Okay. See, this is another figure, another diagram of the pH meter. In this meter, this is also pH meter. In this pH meter, there is an electrode, that is pH electrode. If we put the pH electrode inside a solution, then the pH reading will be displayed on the screen of the pH meter. Okay. So pH is the universal indicator. See, this is the pH scale. Neutral, pH 7, less than 7, acidic, greater than 7, alkaline or basic. See, water is a neutral solution, so the pH of water is 7. And gastric juice, which is present inside our stomach, that is hydrochloric acid. So hydrochloric acid has a pH of 1. Then milk of magnesia, that is magnesium hydroxide, it has a pH range of 10. And the strong base like sodium hydroxide or potassium hydroxide, they have a pH of 13, 14. Soap has a pH of 12. Baking soda has a pH of 9. So these are some of the examples. This is a table, summarized table of color change of different indicators. We have read about the natural indicator, about the man-made indicator. Another type of indicator is olfactory indicators. These indicators work based on smell. When we put the olfactory indicator, some of the examples of olfactory indicators like onion, vanilla, clove oil. When I put the olfactory indicator like onion inside an acid, the smell remains the same. But if we put the olfactory indicator inside a basic solution, inside a base, the smell disappears. So the smell changes. That's why it is known as olfactory indicator. So when we put the olfactory indicator like vanilla, clove oil inside an acid, the smell remains the same. If we put the indicator inside a base, the smell disappears. Now let us summarize. The red litmus, its original color is red. In acidic solution, the color will not be changed, remains same. But in basic solution, the red litmus turns to blue. And in neutral solution, it remains the same red. And then blue litmus, the original color is blue. In neutral solution, remains blue. But in acid, the blue litmus turns to red. But in base, the blue litmus remains blue. No change is there. Talking about the turmeric, original color is yellow. In neutral, no change. In acid, no change. But in basic solution, the turmeric changed to reddish brown color. Talking about the red cabbage juice, its original color is purple. In acidic solution, the color changes to reddish, and in the basic solution, the color changes to greenish yellow. And talking about the China rose indicator or hibiscus, the China rose indicator is in general light pink in color. In neutral, it remains light pink. In acidic solution, it changes to deep pink or magenta. In basic solution, it is changes to green. Now talking about the artificial indicators, man-made indicators, phenolphthalein and methylene. Phenolphthalein is colorless in general. In neutral solution, it remains colorless. In acidic solution, phenolphthalein remains colorless. In basic solution of a pH 8 to 10, it changes to pink color. But in highly basic solution like 13, pH 14, it again changes to colorless. Okay. Talking about the methyl orange, it is orange in color in general. In acidic solution, it changes from red to orange of a pH range between 4.4, but after 4.4, greater than 4.4, the color changes to yellow. In neutral solution, the color of the methyl is yellow, and in basic solution or alkali solution of any pH, the color of methyl is yellow. Talking about the olfactory indicators, onion, vanilla, clove extract. The color in acidic solution, the smell doesn't change in acidic solution, but the smell vanishes, disappears in basic solution. So this was all about the indicators. Talking about the importance of indicators, the indicators help us to identify the acids and bases. The indicators are used in laboratory experiments. They help in titration, acid-base titration. Also, the indicators are used in daily life, like testing soil, testing water, testing the pH of water, testing the pH of soil. So these are the importance of indicators. So this was all about the indicators.